These three standards are the ones more frequently cited in passivation procedures in the pharmaceutical industry. We have first the ASTM A380, that is the standard for cleaning, descaling, and passivation of equipment and systems. Then we have the ASTM A967, that is the passivation of stainless steel parts. And the last one is the ASME standard for bioprocess equipment. This standard has a small section about passivation. A critical thing that we need to understand is that meeting a standard does not equal a corrosion or contamination-free system or equipment. In the following slides, I will give you some examples of how an elementary understanding of the standard and chemistry leads to flaws. We need to keep in mind that some of the limitations of the standards, such as they are difficult to understand, they are for a public familiar with the topic. The passivation conditions on them do not apply to all the circumstances. Meeting the standard does not equal to meet the project goals. Standards provide a good starting point. However, the passivation methods and conditions available to the public have limitations in the following circumstances. Use equipment. Equipment and systems that are not manufactured with the best practices, and metal with specific contaminants. On the other hand, proprietary passivation chemistry may not provide enough evidence about the chemicals involved and the quantitative control of passivation. Tubing and fittings used for pharmaceutical piping, especially if they are electropolished, will pass a water break, ferroxyl, copper sulfate, or potassium ferricyanide nitric acid tests without any cleaning or passivation. The tubing sample on the right is without a question a positive ferroxyl test. On the left, there is a sample of electropolished metal without cleaning or passivation. However, in this circumstance, the ferroxyl test gives a false negative result. The amount of organic matter in sanitary tubing and fittings is minimum, but other stainless steel components that are machined or forged can be more contaminated. The machining of stainless steel requires coating fluids. Traces of coating fluids can be found in machined parts. Aluminum oxide is frequently used to polish sanitary stainless steel tubing. The aluminum oxide used to polish stainless steel will be a contaminant in new equipment and piping. ADT8 at alkaline pH has a better ability to remove aluminum than citric acid, caustic, or detergents. Calcium and magnesium compounds that are common in water can leave residues. ADTA solutions can remove calcium and magnesium compounds better than sodium hydroxide and citric acid solutions. In this picture, we can see the built-up scale inside of a pharmaceutical still. This scale is frequently insoluble residues of calcium and magnesium from purified water. Wells require more demanding conditions for passivation than other parts of the metal. More demanding means longer periods of time or more concentrated chemicals. On the picture, we see the ferroxyl blue stains in wells. Those are a failure, though the tubing passed the test. And when I say the tubing, I mean if we only use tubing as a passivation sample, chances are this will pass easily the test, but it's not the case for the wells. An essential part of cleaning and passivation validation is the testing. Unfortunately, there is no perfect test. Each option has advantages and disadvantages. Usually, substantial evidence of passivation will include more than one cleaning and passivation test. The visual inspection is simple and provides an idea of the metal condition, even if you are not a qualified inspector. A rigorous evaluation will require that the inspector be certified in visual inspections. This is a common practice for boroscope inspections. The main limitation of visual inspections is the area that can be reached. A variation of visual inspection is the wipe test. It is straightforward. We use a clean room wipe on the surface. Any stain on the wipe is evidence of incomplete cleanliness. This test is especially useful to identify polishing residues. The inspection with UV light and the riboflavin spray coverage are two variations of visual inspections. UV light, also known as black light, helps to identify organic matter, like the riboflavin, also known as vitamin B2. The riboflavin is a food-grade compound that is very soluble in water. In the testing, we put on the same process condition a vitamin B2 solution through the spray device. Then we inspect with UV light the tank or the equipment that is supposed to be cleaned. If the spray device is working correctly, all the areas to be cleaned should be covered with the riboflavin solution. The idea behind the water test is that a film of oil or grease will separate in drops the water poured on the metal surface. This test is useful if we have a substantial amount of oil or grease on the metal surface. Usually does not work on stainless steel pharmaceutical components. Keep in mind that fittings, valves, tubings are typically cleaned in the factory and protected from the environment. Pipe fitters should use nitrile gloves to handle all the components that they will put together. The ferroxyl test and the potassium ferricyanide nitric acid test are two versions of the same reaction. They are probably the most used tests to confirm the passivation of stainless steel. A positive test detects the free iron on metal surface, forming a blue compound. This test is easy to do at a low cost. Unfortunately, the chemicals are toxic, so a good practice is to perform the test in coupons that are not part of our equipment. It is a pass-fail test. It does not provide evidence about passivation quality, and on electropolished metal does not provide the required sensitivity. The copper sulfate test's advantage and disadvantage are very similar to the ferroxyl test, with the difference that the copper sulfate test is less sensitive. It is quite useful for non-polished stainless steel. The sprite or immersion test is a lab test. It is limited because the results depend on the appreciation of the person doing the test. The UV spectroscopy is the most sophisticated method to measure the passive layer's thickness and composition. It is expensive, destructive, and only few laboratories have the equipment and staff to do and interpret the test. Other spectrophotometric techniques can provide the composition of the surface. We have seen that many of the passivation tests are limited by the accessibility of the surface. An alternative is to measure the iron concentration in the passivation solution using a colorimetric method. Measuring the iron concentration in the cleaning solution is an indirect method because we do not detect the passive layer, but the iron that we remove in the process. I have used this method and it is so sensitive that it can detect a single weld without passivation in hundreds of feet of passivated pipe. This method's main advantage is clear. The result is evidence of passivation of all the surface in contact with the chemicals. I have personally correlated the efficiency of this method with an OG spectrophotometry and the ferroxyl test. This is how we interpret the soluble iron readings. At the beginning of the process, the iron concentration in our solutions increases, but after some time, the readings reach a plateau. The iron concentration does not increase. The concentration of chemicals used to remove the iron are in excess, but despite this excess and the extended contact time, the iron concentration does not change. This moment is the milestone that defines the end of the passivation process. There are many ways to passivate metal. Depending on the size, geometry, and resources, there are different options. These options include immersion. It's very convenient for small parts that can be deep in a tank. Spraying. It's common for tanks with cleaning spraying devices. Recirculation. Recirculation requires a pump and temporary hoses to create a loop. We use this method for pipes and online equipment. Use of paste. Passivation paste is a sticky solution of chemicals that will remain on the metal surface. The picture on the right shows a pipe covered with passivation paste. Use of foam. We also can spray foam, like in the picture. And this is also a viscous solution that will remain on the metal surface, unlike the paste, that will allow the chemicals to be in contact with the metal and the reaction will happen. The chemistry. It is crucial to know what are you putting in your system. We can easily find passivation products. Unfortunately, many of them are nitric acid solutions with traces of toxic heavy metals. If you use a commercial brand, their composition should be known to the user, and their removal should be demonstrated. The drug manufacturer should ensure that the chemical suppliers notifies them of any changes in the chemical formulation. Many passivation chemical suppliers will not provide a specific composition. This makes it difficult to the user to evaluate residues. Nitric acid is a hazardous and toxic substance. My recommendation is to avoid its use as much as possible. Here you can see a portion of the MSDS for nitric acid, with the warning pictograms showing that it is an oxidant, toxic, and a corrosive substance. You can get similar or better results with the right chemistry using food-grade chemicals. Here is a list of the chemicals used to clean stainless steel and their function. Citric acid is a passivation agent. The best performance of citric acid to remove iron happens at pH 3. Citric acid is perfect for removing elemental iron, but not so good to remove iron 2 and 3. We can use an antioxidant like vitamin C or erythrobic acid to reduce the iron 2 and 3 to elemental iron, and then pick it up with citric acid. Iron is the principal contaminant in new stainless steel, but not the only one. Other metals can be removed with the chelation agents such as ADT8 and sodium gluconate. Sodium phosphates are excellent cleaners. Not all the impurities are soluble. To keep particles in suspension, we use surfactants like Twin 80. Sodium and potassium hydroxides will remove organic matter and are excellent for removing oils, fats, and biofilm. Also can adjust the pH and neutralize acids. We can use formic, oxalic, and phosphoric acids to boost the passivation or remove iron oxides. We may find that some chemicals are not available or very expensive in food grade. However, the quality of the chemicals must be documented. Ammonium bifluoride is available in something called electronic grade with a purity of 99%. Ammonium bifluoride with citric acid will generate hydrogen fluoride, the only substance available to dissolve silica. Hydrogen peroxide and peracetic acid are sanitizers and strong oxidants, which can create a better chromium oxide layer. To formulate gel or foams, we can use cosmetic-grade ingredients with no presence of corrosive compounds.